Hi students, today we are going to discuss about the short notes, very important university repeatedly asking question, glycogen storage diseases, right? One small introduction. The definition for glycogen storage disease is metabolic disorders or diseases due to the defect in synthesis and degradation, that is both synthesis and degradation of glycogen, that is glycogenesis and glycogenolysis, mostly glycogenolysis, sometimes glycogenesis defect also coming under the glycogen storage diseases. This is the definition. We have 7 types of glycogen storage diseases. In these 7 we are going to discuss only 5 because it is a 5 mark question. This is enough for this 5 is enough for your 5 mark question. In these 7 types, all are coming under autosomal recessive. This is very important MCQ. We have 3 important points for this glycogen storage diseases in the introductory point. Number 1, we have hepatic form, myopathic form, and one special form, that is both, either both myopathic and hepato forms. Okay, so hepatic form of glycogen storage disorders are type 1, type 3, type 4 and type 6. For MCQ only I am giving. Myopathic forms are type 5 and type 7, whereas type 2 is the special form, that is general form of both myopathic type and hepatic type. Here what happens is this glycogen gets accumulated in the lysosome of all the cells. What happens is the infant dies before the age of 2 years due to the cardiac involvement or cardiac failure. Gets deposited in heart also and they will go for death. So going to the glycogen storage diseases, you should go through the pathway or cycle of glycogen metabolism. So how it will start from glycogen, glucose, how the glycogen synthesis, that is glycogenesis will occur. From the glycogen, how the glycogenolysis occurs. In these 2 reactions, that is glycogenesis and glycogenolysis, what are all the enzymes involved? Then only it will be easy for you while reading and understanding the concept. For this enzyme deficiency leads to this disease, or for this disease this is the enzyme defect. Like that it will be easy for you, right? Bye. Actually some 7 or 8 diseases are there. I am selecting only 4 or 5 diseases, that is enough for your 5 mark question, right? The disease means the glycogen not able to metabolize. The glycogen will be accumulated in some areas, usually liver, muscle, brain, intestine, like that. That is known as glycogen storage diseases. They are having some scientist name. The disease starts with scientist name. First one is type 1A. 1A means 1B is there but not significant. 1A is very important. Von Gate disease, enzyme defect is usually name of the disease. After that you should mention about the enzyme involved in the disorder. What is the enzyme defect? Third one is due to this enzyme defect, what are all the clinical manifestations the patient is having? These 3 is enough for you. Usually for other diseases you have to mention extra this one. Laboratory investigations, how will you manage, like that. But for glycogen storage disorders, maximum you have to present name of type and name of the disease, enzyme affected, clinical manifestation, that is enough. Okay. So first one is type 1A, name is von Gate disease. Glucose 6-phosphatase, the enzyme defect, that is absent of enzyme or deficient of enzyme. Third one is actually clinical manifestation. We have uploaded one separate video for this von Gate disease, that is why already discussed. Okay. Second disease is Pompe's disease or Pompe's disease type 2. Here there is no AB. The enzyme defect is lysosomal oxidase and acid maltase. Lysosome means it will affect all cells, lysosomes in all cells. That is why lysosomal oxidase. Other name is acid maltase. Here the clinical manifestation is involvement of heart, liver and muscle. Usually in muscle means for babies or infants myopathy. Adult or old age group means muscular dystrophies. You should know the difference between the glycogen involvement in infants, glycogen involvement in adult. Right? There will be some difference. Myopathy, muscular dystrophy. So here accumulation of glycogen in heart, accumulation of glycogen in liver, accumulation of glycogen in muscle produces before 2 years. Maximum the age for the baby is 2 only. Before 2 the baby will expire. Because of this all the involvement of 3 organs. Next one is type 3. 3 we are having A and B. The A is important, Cory's disease. C-O-R-A, Cory's disease. Here the enzyme name is D-branching enzyme. During the glycogen metabolism, we have 2 important enzymes, glycogenesis branching enzyme and glycogenolysis debranching enzyme. Both are producing some diseases. Due to the defect of debranching enzyme, the name, type is 3A and the name is Cory's disease, glycogen storage disease. Here the problem is accumulation of limit dextrin. Limit dextrin is one of the important protein for your glycogenesis. That is also defective. Accumulation of limit dextrin and the liver and muscle also affected. Right? In Cory's disease. Next one is type 4. Here and only one. Type 5, 6 also only one. There is no A and B. Right? Type 4 name is Anderson. That is Anderson's disease. Enzyme name is here debranching. This is branching. Actually, I am sorry. Debranching for glycogenolysis, branching for glycogenesis. Okay. Here the problem is hepatosplenomegaly. All the 3 organs are involved. Hepatosplenomegaly and myoglobin is present in urine. Because of the muscle crush injury like muscle rupture, they will be releasing myoglobin in blood. Finally, they will get excreted in urine. Very serious condition, Anderson disease. Next one is McArdle's. McArdle's. Here the problem is muscle phosphorylase, or some books says muscle glycogen phosphorylase. In your MCQ or any university pattern, you should carefully read about the name. Muscle glycogen phosphorylase is the defective enzyme in McArdle's disease. Here, exercise induced pain and spasm, muscle spasm, right? Because the glycogen is not able to metabolize because due to the absence of glucose 6-phosphatase in muscle. If this enzyme is not present in muscle, means glycogen is not able to convert. That is the reason, right? That is why they will get exercise induced spasm or pain. Last one is type 6, Hers disease, H-E-R-S. The scientist name is H-E-R-S, Hers disease. The enzyme defect is liver phosphorylase. They are having hepatomegaly because of the accumulation of glycogen, right? So, for your 5-mark question, glycogen storage disease, you have to mention 4 or 5 diseases, or sometimes very specifically they will be asking, mention any 2 glycogen disorder like that. For that also, this 2 is enough with the enzyme defect, with the clinical manifestation. Okay. Hi. Next, so the name is Van Dyke disease, that is type 1A. It is autosomal recessive. All are very, very important MCQ point. What is the defect in Van Dyke disease? Accumulation of both normal and abnormal glycogen. Mostly abnormal glycogen. Sometimes normal glycogen also, without metabolism, it will get accumulated in liver, kidney and intestine. The enzyme name is very, very important. Defect of von Geirkes disease is glucose 6 phosphatase. Defect of von Geirkes disease is glucose 6 phosphatase. If it is present in one organ means it will convert the glucose 6 phosphate into glucose. If there is no glucose 6 phosphatase means it will be not be converted into glucose 6 phosphate into glucose. Okay. So glucose 6 phosphatase is the enzyme name. Now we are going to see the clinical manifestations because of this deficiency of this enzyme. Glucose 6 phosphatase is not there means what are all the problems the patient is having, right? Now one is hypoglycemia, fasting hypoglycemia. This is otherwise known as because the whole night the glucose 6 phosphate is not able to convert into glucose. The patient is on hypoglycemia throughout the fast. Second one is lactic acidosis. I will tell you the explanation or reason for these causes. First you should know about the name of the clinical manifestation. Second is lactic acidosis. Third is hyperlipidemia and also hypertriglyceridemia. Fourth one is hyperuricemia. How the uric acid will get accumulated in this von Geirkes disease. Hyperuricemia, one of the important manifestation for von Geirkes disease. The last one is megaly, hepatomegaly and renomegaly. So hepatomegaly leads to cirrhosis and renomegaly leads. So these are all the clinical manifestations of von Geirkes disease. Now we will see one by one. Hypoglycemia due to the defect of glucose 6 phosphatase, right? The problem is glucose 6 phosphate is not able to convert it into glucose, okay, in liver. Second one is this leads to accumulation of glucose phosphate, 6 phosphate in the blood. So this glucose 6 phosphate undergoes glycolysis in muscles, produces finally pyruvate. Glycolysis means end product is pyruvate. In this situation, this pyruvate is converted into lactate because in the muscle it happens in the muscle. So this pyruvate is converted into lactate. This lactate is usual situations. What will happen is enters into the liver and they will go for gluconeogenesis. Here the problem with the help of lactate dehydrogenase which is present in the liver. Here the problem is it will be present in the muscle and also blood. So they will get going on accumulated in blood and muscle, and lactic acid production will be more means lactic acidosis. So first point is hypoglycemia, second one is lactic acidosis. Third point, how this lipidemia and triglyceridemia. Because of this accumulated glucose 6 phosphate, glycolysis is increased whereas gluconeogenesis is decreased. You got this point? Glucose 6 phosphate is available enormously. So this glucose 6 phosphate enters into glycolysis. So glycolysis will be stimulated. Simultaneously gluconeogenesis is decreased. Right? So leads to increased production of NADH, NADPH, glycerol and acetyl-CoA. So from the glycerol, TAG synthesis will be increased. From the excess of acetyl-CoA, ketone bodies is increased. From the NADPH, fatty is increased. Like that, all the lipid compounds will be increased in this disease. That is why hypertriglyceridemia or hyperlipidemia. Okay. The next one is hyperuricemia. This excess glucose 6 phosphate diverted into HMP shunt pathway. So, hyperuricemia means excess glucose 6 phosphate enters into pentose pathway, that is HMP shunt pathway, where this glucose 6 phosphate is converted into purine nucleotides and finally produces uric acid. So excess of glucose 6 phosphate means excess of uric acid leads to hyperuricemia. The last one is deposition of both normal and abnormal glycogen in liver and kidney produces hepatomegaly and renal. Okay, so these are all the clinical manifestations. Finally, two important points. How will you investigate this case? Enzyme study in blood. You have to measure all the enzymes in blood. Second one is go for genetic study. These two are the laboratory investigations for von Geirkes disease. And how will you manage? Nocturnal gastric infusion, that is continuous infusion of glucose for the whole night, 12 hours of the whole night. Then only they will get continuous glucose supply. Early morning no hypoglycemia, otherwise they will go for fasting hypoglycemia, right? Continuous infusion of